All right, let's learn how to draw and annotate how an impulse crosses a synapse. You'll have to pardon some of the spare noises because I am recording this in the hall at school, period. Now, synapses can be in a lot of different places. They can be in between two neurons, so sending a signal from one neuron to another. A synapse could also be between a neuron and a receptor, kind of like your eye or a taste bud or something that detects something else. And a synapse could also be between a neuron and a muscle cell, or a neuron and some other effector. And an effector is something that responds to a stimulus. So think about how if you smell some nice freshly baked cookies or a pie or a pizza or something like that, you might start to salivate. Those salivary glands are an effector because they are responding to the stimulus. Okay, you just need to be able to draw, though, a synapse between two different neurons. All right, I would pause the video right now and make a note that synapses can be between these three different places, but we are just going to draw it in between these neurons. And we're back. So let's start by drawing ourselves the end of a neuron. We call this the presynaptic neuron because it is before the synapse. And then we're going to leave a little bit of a space, and then we're going to kind of create like a cup for that end of the synapse. And this is going to be our postsynapse neuron. So the impulse is going to be traveling down this way through the presynaptic neuron. Now, in the membrane of this presynaptic neuron is a channel protein, and these are calcium ion channels. They are voltage gated, which means that when the impulse reaches them, they change because of the voltage and open up. This allows the calcium ions to flow into the presynapse neuron. So we can call this number 1 right here, and this is number 2 right here. So calcium flows into this synapse here. So we get a bunch of calcium ions out here. This is passive transport because there's a high concentration of calcium ions on the outside. Now, inside of this presynaptic neuron are a bunch of vesicles, and remember, these are just membranes that surround something. And what they're surrounding is a whole bunch of a molecule called a neurotransmitter. So these are neurotransmitter filled vesicles. Okay, so once the calcium attaches to these neurotransmitter-filled vesicles, that is a signal for them to do exocytosis. They're going to merge with this membrane here. Oh no, that's too much of an eraser. Okay, so let's redraw a little bit of that here. They're going to merge with this membrane here, and they're going to dump their neurotransmitters out here into the synapse. Now the synapse is filled with all kinds of little receptors here that perfectly fit. So these neurotransmitters then attach, this is number 4, attached to these receptors. Now, right next to these receptors are what number 5 is, which is some sodium channels. And these sodium channels are going to open up. Now, there's a high concentration of sodium outside of the neuron, a low concentration of sodium inside the neuron. So the sodium rushes in, and we know from our action potential that when sodium rushes in, the voltage of the inside of the membrane becomes positive, and that starts the impulse happening. Number six is that there are enzymes that sit inside of the synapse, the gap in between the neurons, and those degrade or break down the neurotransmitters so that the signal doesn't keep going. Those neurotransmitters get reabsorbed. Neurotransmitters get reabsorbed by the presynaptic neuron. And the whole process can start again. So once again, pay attention to the things that I said, not just the things that I wrote. Otherwise, you're missing some real key important information. All right. Make sure to watch the other two videos as well. And that's it.